because of this injury that has occurred. One thing that I also start by pointing out here that will become a recurring theme throughout my talk is there are sex differences in traumatic brain injuries. So you'll see in this young age group, males are more likely than females, so young boys are more likely than young girls to sustain a TBI that sends them to the ER. It's not 2-to-1 or anything like that, but there are some sex differences that may come into play. Still can't use the keyboard. So there has been some work in humans tracking long-term behavioral outcomes associated with early life brain injury. And some of these are subtle, but they do emerge when you look at the population level or epidemiologically. So early life TBI does increase rates of psychiatric disorders as well as social difficulties when looked at in childhood. And that's in both males and females. But when you start parsing the data by sex, you see there are some sex-specific effects behaviorally. So when you look at males in particular, you see that boys tend to have poorer learning and verbal outcomes than females do after an early life TBI in a school setting in particular. In addition, there are externalizing problems, and what we mean by that are things like ADHD, things like substance use, as well as longer-term cognitive and social deficits. Those tend to be more common in males than in females after an early life brain injury. On the flip side, internalizing problems, things like mood dysregulation, risk for depression, anxiety disorders, tend to be more common in females. One thing I don't have on here, but I saw recently was also risk for chronic headaches, like migraine, tend to be more common in females that have sustained early life brain injuries. And the mechanisms through which many of these things are occurring really have not been investigated. So there are a few things we know, but a lot that we don't know. So my lab is really interested in the big how. How does pediatric TBI potentially program this long-term risk for these behavioral complications? So I'm going to go through just an overview of some of the mechanisms we're interested in my lab. And then I'm going to walk through some of our projects on pediatric brain injury and kind of where we are in them. So one thing my lab is really interested in and has been basically since it opened is the role of immune cells in programming neurotypical development. And right around the time that I was a postdoc, this field kind of blew up. So microglia, which are the innate immune cells of the brain, are about 10% of our brain cells, and it has become increasingly clear that they regulate many normal processes of brain patterning. So our brain grows and it refines itself over the course of your brain's, development. So really from conception until really you're an adult. And that comprises many different processes. So cell genesis, cell death, axon pathfinding, myelination, and then synaptic refinement, both synapses growing as well as synapses being pruned. And what has emerged largely through rodent research, but some research in other animal models, as well as a burgeoning research in human stem cells and a little bit of actual human imaging and things like that, is that microglia contribute to these processes. They largely do this by secreting factors that can support these processes, so growth factors, for example, but they also engage in this process called phagocytosis, where they engulf and digest cells, so excess cells that need to be pruned, but also synapses that are considered, extraneous or weak, for example. Microglia are also necessary for behavioral development. This has been investigated considerably less, but this is some work that my lab has done and some other labs have also looked at. But we've done some work where we actually got rid of microglia from the brain during the early postnatal period and found that when microglia weren't around, there were dramatic changes in a variety of behaviors, those related to mood, including changes in anxiety-like behaviors, fear responses, as well as some changes in things like cognition. So microglia regulate normal brain development as well as behavioral development. And that made us really interested, and other people, in whether they could contribute to changes in response to early life perturbations. My lab is also interested in another type of innate immune cell that gets a lot less attention out there in the world, and those are called mast cells. A lot of people have gotten a little interested in them on like TikTok and the internet and things like that. So you might have heard of them, especially after the COVID pandemic. But these are cells that are involved in allergic responses. So things like allergy, asthma, as well as our response to itch. So they're in our skin and our airway, and that's where they receive the most attention. But they're also in the brain in relatively small numbers, but they're very powerful cells. And they undergo this process called degranulation, where basically they're activated and they dump huge amounts of inflammatory mediators into their surrounding tissues. So many of the same things that other immune cells make, but also interesting things like neurotransmitters, serotonin and histamine. Interestingly, they can crosstalk with microglia, and some people have looked at them in other injury conditions, like in adulthood, as well as in other neurodegenerative states. But they're very interesting because they can regulate things like the blood-brain barrier, as well as immune trafficking. They really haven't been studied much in the developing brain, although my lab has done some work on that. We have implicated them in some of our other work in behavioral development, as well as done some research on them in the context of early life inflammatory challenges. And so we know that they contribute to the development or changes in social and emotional behavior in the context of prenatal immune insults. So I'm just kind of priming you to be interested in them, hopefully, in the context of brain injury. So given that we know that these immune cells are important, they're in the brain during development, we are pretty interested in understanding whether if we perturb them potentially with insults such as a brain injury during neurodevelopment, might that make the brain particularly vulnerable to behavioral changes? If so, we want to figure out how that's the case. And we're really interested in understanding whether all early life insults are similar or whether there are unique signatures to different developmental insults. So I'm going to start by talking about brain injury, and then I'm going to pivot to another sort of perturbation that we're also interested in to do a little kind of comparing and contrasting. And then think about whether insults experienced like in tandem or alone are unique. So I'll first talk about our preclinical model of pediatric TBI, which wouldn't have been started without support from the CBI, and then pivot a little bit to talking about some work on early life stress and how it interacts with TBI. So I think a lot of you in the room use animal models, so I'm probably preaching to the choir. But given that not everybody thinks about the utility of animal models, I just want to emphasize the power of using animal models in the brain injury space, or really any studies where we're trying to think about complex behavior. So brain injury can be very complicated, and the benefits for us in using an animal model are really that we can control the incredibly variable circumstances of a brain injury, the severity, the timing, et cetera, in order to really get into the nitty-gritty and discover how, when, where in the brain injury is affecting brain development and behavioral outcomes. And yes, a mouse or a rat is not the same as a human, right? It's not a tiny human, but there are so many similarities in the trajectory of neurodevelopment that it allows us to make really good comparisons between how brain injury in a kid could be affecting things like myelination or cell birth or synaptic pruning that really is very hard to study in people, so that we're able to discover hopefully new mechanisms that are underlying brain injury pathology and then translate those new discoveries into the clinic. So this is just a diagram showing that there are very similar trajectories to the processes of brain development between a human and a rodent. And that allows us to map on both our model building to a rodent from a person, and then hopefully back translate those things back and forth between our animal models to clinical applications. So how do we model brain injury in our rodents? Well, I know many of you in the room that do TBI work do it different ways, but this is the way that we do our brain injury in our neonatal rats. And this is collaborative work that we started with Nikki Cochran's lab a number of years ago with a CBI pilot grant. And this is an injury called lateral fluid percussion, which is essentially applying a fluid pulse to the surface of a neonatal rat's brain. It essentially causes both a localized as well as a diffuse brain injury. And we have found that in our animals, which are at postnatal day 15, which is roughly similar to kind of like a toddler to preschooler age, neurodevelopmentally in a kid, this is actually a relatively mild injury, so mild to moderate. So we see, compared to our sham animals, our traumatic brain injured animals do spend more time before they kind of wake up after the injury, so it shows that they were in fact injured. However, if you just look at the animals, even hours after the injury, certainly days after the injury, to an untrained observer, I mean, they have like a little boo-boo on their skin, right? You know, they have like little wound clip on their skin. But if you didn't see that, you wouldn't know that they had had a brain injury. Like they're just waltzing around, like nothing happened to them. To the untrained observer, they don't have overt physical impairment. So we think that this is a good model to those 75% of brain injuries that kids are getting. They're maybe going to the ER because their mom was scared, but they're going home pretty soon after and kind of resuming their quote unquote daily life back in their cage with their moms. They're eating normally, those kinds of things, behaving relatively normally. So we think it's a great model. It's not as if like they're needing huge amounts of supportive care, the equivalent of being in the hospital type thing. So the question really then is, what are their sort of acute outcomes and what are their long-term behavioral outcomes? So the central questions of our work are here. What are the acute inflammatory responses? As I told you, we're really interested in the neuroimmune cells in the brain. What are the sort of midterm neurodevelopmental consequences, those things that we think neuroimmune cells are regulating? And then what are those long-term behavioral consequences of this early brain injury? Today, I'll tell you about one, I'll tell you about three, and you'll have to stay tuned for number two, which is a lot of work. So from now on, superstar of all this is going to be Dr. Marissa Smales right there. Sorry, you have to look at Marissa. She's a postdoc in my lab who's done a huge amount of work on this project. And then the next project I'll tell you about too. So I'll first tell you about our sort of acute histology endpoints, which really have been focused on determining whether there's regional specificity to the immune cells' response to the brain injury and which immune cells are sort of preferentially activated, if so. So we focused on a few brain regions, just to not let it balloon out of control. We first focused on the cortex right around where we actually have this injury occur. And we focused on two other limbic brain regions, the hippocampus, as well as the prefrontal cortex, which are regions that are involved in a lot of those behaviors that I told you are impacted in humans. So mood, social, cognitive related behaviors to see whether we have inflammation occurring there. So first of all, when we look at just gross immune cell kind of coverage near the injury, and then at those kind of more distal areas associated with the behaviors we're interested in, we see that TBI increases staining for microglia, as well as another immunocompetent cell, astrocytes, right around the injury, about 3 days after, but not farther away in the prefrontal cortex and the hippocampus. So this is telling us that this is probably a pretty mild injury. So we do see some inflammation near the injury, but not farther away. Interestingly, when we look at a marker of microglia phagocytic activation, so remember I was talking about how microglia sort of gobble up material, things like dying cells or synaptic material. When we look at that marker, CD68, and we actually do this with a neat program called MRIS, where you can actually look at this marker within microglia, we see that not only near the site of injury, but also much farther away at the prefrontal cortex, as well as in the hippocampus, we see elevations in the CD68 marker. So while we might not see overall increases in microglia staining farther away, we do see increases probably in this particular activation of microglia. You'll note some places I have the sexes split out. That's where we saw a sex difference between males and females. And in effect, if I don't have them split up, then the males and females are kind of lumped together. The triangles are females and the squares are males. So in this case, we see that the females have a more profound elevation in their phagocytic activity in the hippocampus, which may have implications for those midterm neurodevelopmental changes that we just haven't had an opportunity to look at yet, or are in the middle of looking at, I guess I would say. When we looked at mast cells, so those interesting immune cells that everybody ignores, we found, in fact, that TBI led to a significant increase, both in males and females, in response. And in particular, when we look at the degranulation, so the activation of those mast cells, we see a strong TBI effect. So we're really interested in pursuing that and seeing what the mast cells are doing and whether they're potentially either leading to the increased microglia activation and or regulating things like blood-brain barrier permeability, which we know is kind of one of their specialties. So the next experiments that Marissa did were kind of behemoths. They were two really big behavioral experiments looking at the long-term behavioral consequences of pediatric TBI so that we could zero in on which behavioral domains may be most affected in this model, and we could zero in potentially on the mechanisms that are most interesting. I'm not going to tell you about all of these. We'd be here for a while, but Marissa can certainly tell you about them all if you want to know, or you can read her paper that she's almost done writing. No pressure, Marissa. I'm going to tell you about just a few that I think are interesting, particularly interesting, and maybe ones that you don't always hear about. One's juvenile social play. I like this one because mice don't do it, rats do it. It's like a great justification for why we study rats. I'll tell you about social behavior. I'll tell you about some cognitive behavior, and then I'll tell you about a task that we've done in collaboration with the Vonderhaar Lab. So I'm gonna show you this video, social play behavior, because it's just like, it'll warm your heart on Friday morning. So social play is a really analogous behavior to human childhood play. Mice don't do it, rats are better, guys, sorry. And it's a highly motivated behavior. It's essentially like rough and tumble playground play that you see in any school playground. So it mostly consists of wrestling, chasing, pinning, highly gregarious, highly rewarding, and it's only concentrated to the juvenile period. So when animals reach sexual maturity, they more or less stop doing this. They're adorable. It's a nuisance for trainees in the lab to score because you have to hand score it, but I mean, I like to think at least it's fun. So what happens to this behavior after pediatric TBI? Well, it's impaired. So in our animals that received a TBI, we see decreases in play frequency relative to our naive animals and to our sham animals. So this is kind of similar to what the epidemiological literature would suggest, is that there's social changes, but really in this motivated behavior, we see this decrease, which is quite interesting. When we look in adulthood at a open-ended social interaction task, we also see more long-term changes in social behavior. So when a rat is paired with just like a new friend, essentially, that they've never met before, and we just code how much time they spend interacting with that friend, we find that the TBI animals spend significantly less time interacting actively, so pursuing active interaction with that friend, and they actually spend more time avoiding that friend. So it's like if that new guy comes up to you, you're like, yeah, thanks, and you walk away. So they are both engaging in social avoidance as well as less active social interaction. So this tells us months after this relatively mild TBI, we see social behavior deficits. So we think that social behavior is likely a domain that is particularly vulnerable to this pediatric TBI, and we're pretty interested in pursuing the neural circuits underlying social behavior and the mechanisms through which it might be altered. I also looked at cognitive behavior, so this kind of short-term declarative memory through the novel object recognition task. So you give them two objects and have them look at them, and then you switch one object a little bit later. And most rodents prefer novelty, so if they remember the objects they saw before, if you give them a new object, they should prefer this new object because it's interesting and new, shiny and new. But the TBI animals actually spend less time, so their discrimination index is lower. They spend less time looking at the new object, suggesting that their short-term declarative memory is impaired. Finally, like I said, in collaboration with Cole Vonderhaar's lab and Chris Martins in his lab, we did a new, very challenging to us task, which is called the rodent gambling task. So this is a task to assess risky decision-making. It's kind of similar to people who do sort of neuropsychology to the Iowa gambling task, where animals is an operant task where they have to essentially learn to press a lever. Is it nose poke? Which one is it, guys? I don't know. Nose poke? Okay, thank you, Vonderhaar lab member. They have to nose poke to get a sugar pellet reward, and there are different levels of reward that they can get at different rates, essentially. And some are more risky. So if they go to that particular choice, if they choose it and win, they can get a really high reward. But then if they choose it and lose, then they get like a timeout that's really long. So it's like a risky choice. There are other ones that are like a good bet, essentially. So the individual reward they get that particular time is lower, but the rate at which they can get it is higher because their timeout, if they lose, is lower, essentially. So over time, they have to sort of make the kind of cost benefit analysis and figure out which is like the optimal choice. Like, yeah, each reward I get is lower, but I can get more over time, right? And so it's a sort of decision-making learning task. So here it's kind of broken down on which tasks are more risky and which choice is most optimal based on that reward rate. And as you can see, 2 is the optimal choice. And you can see that our TBI animals on this task, and this is literally like 4 months after they got this TBI. Our TBI animals make much less of the optimal choice relative to our naives and our shams. And if you look down here at the risky choice, you can see that our TBI animals are making much more of the risky choice relative to the shams and the naives. So there's this shift towards risky decision making, and maybe it is that the sort of salience of the high reward is higher. Maybe it's there are a lot of like competing things that could be going on, but we're really excited about this data and what it might sort of indicate about the neurodevelopment of this potential system involved in sort of decision making. My little animations. So interim summary, this model of pediatric TBI we show demonstrates acute activation of neuroimmune cells and increases in those phagocytic microglia and mast cells in limbic brain regions, as well as long-lasting social behavior deficits and adult cognitive deficits and risky decision making. So we're pretty excited about getting into, like I said, that number 2, those neurodevelopmental changes that could be mediating the long-term behavioral outcomes. So our ongoing work that's being spearheaded by another postdoc in my lab, Courtney Dye, and one of my lab techs, Miranda McDonald, is assessing whether if we pharmacologically inhibit mast cells at the time of brain injury, whether we can prevent behavioral changes and or neuroimmune activation. So this is something we're doing via CSF infusion of a mast cell inhibitor called cromolyn. This is just our preliminary data showing it's working at the time of injury a few days after we see significant increase or decreases in the number of degranulated mast cells. So we're really excited with upcoming behavioral cohorts to see whether we can rescue things like those social behavior changes, cognition changes and whatnot. And currently, we're also assessing whether TBI impacts microglia interactions with like developing circuits in the hippocampus in particular, given those increases in phagocytosis we saw. So now I'm going to switch gears and talk a little bit about early life stress and our second pediatric TBI project. So pediatric TBI is a problem, but it's not the only problem for kids out there, unfortunately. So another highly prevalent public health concern that increases risk for negative health outcomes like behavioral outcomes is early life stress. So I'm sure you've heard of adverse childhood experiences. So these are a variety of negative childhood events that can have pervasive, lasting effects on a variety of health outcomes. So I'm a neuroscientist, so I'm focused on the effects on brain and behavior, but these effects of ACEs are wide-reaching on the body. So all-cause mortality is increased if you've experienced a variety of early life adverse childhood experiences. It's not just focused on brain development and psychiatric dysfunction, for example. However, individuals, in particular those who have experienced 4 or more adverse childhood experiences, and these are just things like parental neglect. I mean, they certainly can be things like abuse. They can be things like losing a parent, food instability, these kinds of things. They can increase risk for things like depression, suicide attempts, substance abuse across the lifespan. So they dramatically increase the risk for a lot of the same things that we're thinking about when we're thinking about pediatric brain injury as well. And almost 2/3 or over 2/3 of individuals have experienced one ACE, but it's really individuals that are getting compounding ACEs that really move into the high risk category. So here it says about 1/8 of individuals are in that really high risk category of experiencing more than 4 ACEs. So we're pretty interested in trying to model some of these ACEs in preclinical setting and try to understand mechanisms through which they're impacting brain development. So how do we do this in a rodent? There are a few different models out there. This is the one we use, which is called maternal separation. And it essentially is a model of parental neglect. So we have, mother rats. We actually have another project with mice, and we do something pretty similar. But the mother rat typically takes care of her pups very closely for the first 14 to 21 days. I mean, the last week or so, the pups are pretty mobile, so they get independent. But the first 14 days, they're pretty dependent upon mom. So we separate that strong bond for about 4 hours a day. We take the pups away from mom. We keep them warm and cozy by putting them in little solo cups and then sticking them in a chicken incubator that we bought from hatchingtime.com. They're like, do you want the egg turner? We're like, no, we're not hatching eggs. But they didn't understand. So we take the pups, we put them in solo cups in an incubator, keep them warm and safe, but they miss their mom for 4 hours a day. This disrupts the maternal caregiving during that time, and it does elevate stress hormones in the pups. Then we reunite them, mom takes care of them, all that, they get fed, all that jazz. But we do this daily for 14 days. And this is a very well validated model. It's been used for decades. We didn't make it up. It's known to elicit anxiety behavior in the offspring in adulthood, decrease things like their cognitive flexibility and working memory. It's known to perturb their basal stress hormones throughout their lives. And importantly, if you're a person who likes neuroimmunology, it elicits neuroinflammation, particularly via microglia. So don't take my word for it. And we wanted to make sure that it elicited neuroinflammation in our hands. So we found that maternal separation stress does indeed lead to neuroinflammation in one of our brain regions of interest, the hippocampus. So we see increases in microglia staining in several subregions of the hippocampus right when the animals are weaned. And we also see increases in some markers for that microglia phagocytosis, the gobbling up that we're interested in. So these are just a few genes related to phagocytosis, TYRBP and C1q. We see those elevated in the hippocampus of pups right at the time that they're weaned. So we know that the stress, this early life stress alone does in fact elicit neuroinflammation and phagocytic changes in the hippocampus. We previously asked some questions about whether these microglia are necessary to program later life behavioral changes in response to maternal separation stress. This is a really simple experiment where we just got rid of the microglia using a drug called liposomal clodronate at the time of the stress itself. This was done by a previous grad student in my lab, Lars Nelson. And what we found is that stressed animals show anxiety-like behavior on a task called open field. So they spend less time in the center of this open field. So they're less brave. They don't go into the center of the field as much because it's scary. And animals that experienced microglia depletion at the time of stress have their bravery restored, essentially. So they look no different from the control reared animals. So we know that this early life stress, the effects on anxiety-like behavior do seem to have a microglia component to them, which is consistent with our hypothesis. And that led us to be really interested, given that we saw kind of similar things going on with our pediatric TBI, where we saw increased microglia and phagocytosis in the hippocampus, and our early life stress project, where we saw increased microglia and phagocytosis in the hippocampus, to try to figure out whether these two perturbations had any relevance to one another, and whether, if we put them together, whether they might synergize. And that wasn't just, idle curiosity. There's also good epidemiological reasons to wonder whether those two things may synergize. So out there in the literature, there is at least a little bit of evidence that early life stress and TBI can interact. So a history of ACEs, those adverse childhood experiences, do predict worse psychiatric outcomes, especially after adult TBIs in military populations. And it should be noted that one of the ACEs out there are physical abuse, right? So those are some of the components to ACEs that are most highly reported. So physical abuse can actually even lead to a TBI. So these things can be directly comorbid. Individuals that are experiencing ACEs may actually sustain a TBI. In addition to that, I talked about the top two causes of TBI in the pediatric population: falls and being like hit by an object like a ball. But the third most common cause of pediatric TBI is actually physical abuse. And in addition to this, the co-occurrence of physical abuse with ACEs has been shown in a few epidemiological studies to predict poorer mental health outcomes. So like not all ACEs are created equal, I guess. So physical abuse in particular is particularly troubling for mental health outcomes. Yet few studies have actually assessed how these two perturbations interact to shape the developing brain. And we felt like that was something that we could do, at least from a preclinical side of things, to hopefully spur more translational and clinical research. So the first study, again, here's Marissa, and the first study that she did was use this maternal separation paradigm that we had established and combined it with our pediatric TBI model, the lateral fluid percussion injury, and did a behavioral battery to determine which behavioral domains seem to be impacted by these two perturbations when they were combined together. And I'm just going to show you a few of the behaviors again for the sake of time. So I'll show you novel object. I don't know if I actually will. I'll show you open field, maybe. I'll show you social interaction because we talked about social behavior before. I'll show you stress hormones, and I think that might be it. Okay, I guess I am showing you novel object. So when we did novel object, interestingly, with all of these things, we were kind of predicting like the simple thing, like, stress plus TBI would make things, doubly bad, right? And it has not been that simple. So in the case of the novel object, we again replicated that we had a deficit with pediatric TBI, and we found no additional impact of stress plus TBI together, which is this bar over here where the animals had maternal separation stress plus TBI. So the stress had an effect too, but the two together didn't make it even worse. So maybe there's only so bad you can be at a cognitive behavior, and both stress and the TBI make you impaired on this task. When we looked at social interaction, we actually found kind of an interesting thing where the pediatric TBI led to decreases in active social interaction, which I guess we had seen before, whereas stress plus TBI together actually led to this kind of two-hit effect on passive interaction where the animals just kind of like huddled next to each other more. So this is almost like I think of it, maybe I'm anthropomorphizing it, but I think of it kind of like a personality change where they're kind of like, just like less assertive, you know what I mean? Like they have more of this kind of passive personality when they're in this social space. So they kind of let the other animal be by them, but they don't actively pursue interaction with them. So that'll be an interesting thing to try to tease out. I don't think a lot of work has been done on figuring out the dynamics of sort of active social pursuit versus passive social temperaments in rodents. This is one where we actually found like an additive effect, especially in males. So when we did the anxiety-like test, behavior test, the open field, we found particularly the animals exposed to early life stress plus pediatric TBI showed a dramatic decrease in their open field center time. So they were the ones that were really hesitant to go into the scary center of the open field, indicating anxiety-like behavior that was really potentiated by those two hits together. Females, on the other hand, were a little more complicated, where we saw an anxiety-like phenotype just with the TBI that then somehow was turned around by the maternal separation stress. So a situation where the two hits are pulling in opposite directions, which is like much harder for us to get our heads around. And with these two hits, sometimes you see these things and they're definitely challenging to interpret. Finally, this is a pretty interesting one. We put animals in a water bucket, which they don't like. It's stressful, and that's the point. And then we took blood samples to test their stress hormone levels. And then when we looked at their stress hormone effects, we found dramatically different effects based on their sex. So the maternal separation stress plus TBI, they're these green hash bar ones. And then the blue is our controls. And in our males, we can see that they had a much more dramatic stress hormone response to being stuck in a bucket of water. So they found, at least their hormones found this more stressful. Whereas in our females, the animals that got the two hits actually had a dampened stress hormone response. So this is what we call a latent sex difference or a divergent response to the two hits together. So at baseline, the animals don't look that different, but when you give them these two hits together, males and females responded dramatically differently. So this says something about their endocrine state or their physiology is different, even though males and females experience the exact same things. My background's in neuroendocrinology, so I'm very interested in getting to the bottom of this, but it might suggest that they will respond very differently in threatening situations if their stress hormones are this different. And then finally, Cole's not here, so he can't laugh while I try to explain this. But we did the rodent gambling task again. And this time, we found very different responses in our males and our females. I'm going to just tell you, like, ignore the males. They were kind of messy, and we didn't see as dramatic, clear TBI effects as we did last time around. But I just pulled out the optimal choice and the risky choice on the rodent gambling task. And in this case, the females showed really clear synergistic effects of maternal separation stress plus pediatric TBI on risky decision making. So the two hit guys are these green hash ones again, and we saw the optimal choice with the maternal separation stress plus TBI, the females really decreased in their optimal choice at the very end, particularly of this cohort. And then they showed a real increase in the risky decision making. So in this case, especially the females showed an increase in risky decision making. I'm getting close to the end of my time, so I'm gonna kind of rush through this. The last thing that we've done, and this is really preliminary data, is a pilot study using single nucleus RNA-seq to try to start getting at what some of the mechanisms through which these behavioral effects of TBI and early life stress could be occurring. So like I said, this is a pilot study that we did a little while ago now. We're in the process of doing this again in a fully powered manner, but I just wanted to give you a tantalizing teaser of what's to come. So this is a study in which we did the same idea, two hits of maternal separation stress followed by our injury. And then we dissected the hippocampus 7 days post-injury, prepared samples, and performed single nucleus RNA sequencing really to really focus on the neurons and how neurons were responding to this brain injury. I want to note, just so you're not confused, that this was a considerably milder hit than our current injury. So based in part on this data, we realized we wanted to make our injury a little bit stronger. So this was a previous injury model. And this was done by Michaela Breach in my lab with a lot of help from the Godbout lab from Ethan Goodman and Jonathan Packer. So our hypothesis here, again, was that early life stress would exacerbate TBI's response. This is just our clustering. Just want to emphasize about half of our isolated nuclei were neurons. And I'm really just going to focus on some of our pathway analysis in neurons. We have looked at some of our other cell types too, particularly microglia, but I don't have time to talk about that. So we did find differentially expressed genes in both excitatory and inhibitory neurons by condition. As you'll see, stress seemed to be have stronger effects than the TBI, but I think that's in part because of our weaker TBI we used. We found strong increases. These are heat maps showing increase in yellow and decreased gene expression in different pathways in purple. We found really strong increases in pathways associated with synaptic plasticity by stress. TBI in some cases heightened those. We found opposing effects in a lot of cases by stress and TBI on neuromodulatory pathways like endocannabinoids and opioids, which are pretty interesting for neurodevelopment, and then synaptic plasticity once again. We also found in excitatory neurons strong stress effects on neurohormone signaling, which in some cases like oxytocin and androgen signaling were countered by TBI. With inhibitory neurons, I don't want to dwell too much on this because we found really strong stress effects and very few TBI effects. So again, early life stress strongly affected synaptic plasticity pathways, and TBI really had no effect. And then with inhibitory neurons, we found several effects on growth factor pathways, including VEGF, Liz, with TBI having few effects on those as well. I think I'm going to skip this because I'm out of time. So in conclusion, we think our 2-hit model is really interesting, and that together between our sequencing data and our behavioral data, we have interesting sex-specific effects and cell type-specific effects to pursue in our future mechanistic work. So our next steps are assessing microglia interactions, as well as doing what we did in, oops, excuse me, what did I do? I'm just leaving it. Oh no.
Recording your voice.
I know. Okay, too bad.
And yours too. And mine too.
I would stop it at no time.
Did you hit that audio button?
Okay. In conclusion, did that layout there under
I think we're talking.
no. Okay.
Oh, myself. That's even worse. Seeing the song. I sing to my friends. Recording while I was talking.
That's it. I would just leave the transcription up. You're almost.
It's fine. I know. Well, how do I make it bigger?
That at least got rid of the.
I sent it.
There you go.
Yeah. Okay. Anyway, we're just gonna skip through.
And then I'm like trying
So
to
in
read it.
conclusion, fun time. I really just hope I convinced you that both pediatric brain injury and early life stress are prominent, but often really invisible public health concerns, and that our preclinical models can lend some insight into the mechanisms through which they're programming neurobehavioral development, either alone or in tandem. I think our preclinical studies thus far have given us insight into long-term behavioral consequences, particularly in domains of cognitive function, social function, and mood function, and some underlying pathways through which they may be occurring. We're just scratching the surface on the underlying mechanisms, but we have a lot of work coming, so stay tuned. I really also want to emphasize that greater communication and collaboration between preclinical researchers like me and translational and clinical researchers will be the way that can pave the way to new treatment and prevention strategies. And then I'm just going to finish with My acknowledgements, my awesome lab. Marissa in particular has done most of the work I presented today. Michaela did the single nucleus RNA-seq, a whole army of grad students, techs, undergrads. I have the best lab. Collaborators, I couldn't do this without them. Dr. Kathryn, who got me started on TBI, is my TBI and spirit guide. And then Dr. Gabout and von der Haar, with whom I collaborate really closely on this work. Dr. Lunar and Dr. Barrientos, that are on my R01, and Dr. Espeth in the EFIS core. This work has been funded by my R01, 2 DoD grants, one of which is with Gabout and Kathryn and von der Haar, our big program project. In this day and age, we just have to be thanking our federal funding sources more than ever. But I've had foundation funding, CBI funding, pre-award, and then foundation award too. So thank you for your attention.
Very nice presentation. And it's nice to see it all kind of coming together, seeing it all at one time. And when you think about the behavior you showed, especially in the first part of the talk, correct if I'm wrong, but the rats have more of an antisocial profile behaviorally, but yet have a higher risk taking. Yeah. So when you think about that, does that allow you to target specific neural networks or circuitry that kind of made that you look to see if those specific circuits are affected?
I mean, that's our goal. I think like social decision making is kind of a different domain than decision making, but there are sort of overlapping Venn diagrams. So I mean, we're hoping that when we do our single nucleus RNA-seq, we'll get insights into which neuromodulatory pathways are involved. I mean, there's so many different factors, so many different neurotransmitters and systems that can modulate those behaviors. So I'm hoping that our sequencing data will give us insight into where it'll look first.
And then with the sequencing, the little bit you showed us, is that overall you see, really kind of a stress enhanced number of things associated with plasticity in inhibitory and excitatory neurons. And in your TBI, it was either a suppression or no effect.
Right. Yeah, I mean, some people hypothesize with stress in other models that there's sort of precocious neurodevelopment, that it sort of accelerates neurodevelopment. And so that's one idea that might be going on. And I didn't have time to show the microglia, but there are certain pathways in our microglia in the sequencing that are suppressed. So there may be, microglia are usually kind of a brake on oversprouting of synapses and things like that. So maybe some aspects of microglia dampening neurodevelopment are dysregulated by stress and TBI. So something we want to look at. Yeah.
I'm curious what other brain regions you think might be involved, because you mentioned like decision making and
yes.
behaviors, and I'm not biased at all, but maybe some parts of cortex.
Yeah. So our current sequencing is focused on prefrontal and hippocampus, but we're open to branching out too based on our behavioral domains. But yes, we're looking at prefrontal cortex in our sequencing now, and actually a lot of our other assessments too. Yep.
I was just interested in like the behavior and the sex differences, because in the beginning of the talk and like clinically you kind of talked about how in males and females that experience pediatric TBI, males tend to have more externalizing where females have more internalizing. Yeah. But it looks in like your data with the 2-hit at least that it's almost the opposite. Like you're seeing males are having more anxiety disorders and females were having more risky decision making.
I know.
Which would probably be So I'm so sorry to bring this to your attention.
I know it is interesting.
Do you think maybe like you can explain that as a result of like the 2-hit? Like, is it that in TBI alone, this might be what is seen clinically, but
yeah, I mean, that may be some of it. And I don't think there really hasn't been any human work looking at outcomes, especially in children, as a result of the 2 hits. So that could be part of it. A lot of it, I always suspect there could be some bias on, like, we look for what we expect to, like, we expect girls to have mood dysregulation, so we look for that, and we expect boys to have ADHD, so we look for that, or, I think that diagnostic bias is part of the issue in the clinic too. And that's why I feel like having the preclinical data can maybe push people to like open their eye. I mean, I know it's a rat, but like it also, I think, can help people think a little more broadly about what we should be looking for in kids too. And maybe we have some blind spots based on our, our gender stereotypes. Yeah.
Have you looked at the body weight? I'm like curious because I'm wondering if either of your manipulations or both combined can affect their feeding or their ability to compete for like milk from the mom.
Yeah, we have.
Things like
Marissa can answer that.
Stress, so the stress decreased weight, not a ton, but like it stayed. The TBI decreases it for like a week following, but they recover back to normal after. So the stress effects have outlasted the TBI ones, but it's not like a dramatic change.
in the combined, are they just similar to the
They're
stress?
they're
Yeah.
those ones.
Yeah, because I also think of, I mean, I'm just curious
Mhm.
whether anyone thinks about if feeding, like suckling, is
Yeah.
social, could be considered social to some degree.
Yeah, and I mean, we have looked at the maternal behavior too with the TBI. It's not like dramatically altered, like nothing significant. So. Yeah.
I'm just curious, for your single nucleoside seq data set.
Yeah.
So you show excitatory neuron and inhibitory neuron both focus on the synaptic related gene pathway, at least, right? The very common similar. So I'm just curious, do they have a common gene and then in both excitatory and inhibitory neuron? So it then can be targeted in the future.
I think there was a lot of overlap in the genes, yes. But not, I can't remember the percent similarity.
Yeah, that's a many gene there.
Yeah.
Yes.
Mhm.
Any online questions?
Nope, nothing in the chat. Just go back to the attendance slide real quick.
No problem. All right, thank you all.
Session good.
Yeah, you want
I also want to break it.